Hello, Deepa. We'll get started in about 2 minutes. Okay.
Sure.
Okay. Thank you. Hi, Deepa, are you here?
Yep, I'm here.
Okay. Okay, we'll get started. Welcome everyone to our Progress and Prospects in Biology weekly webinar. Today we have with us Dr. Deepa Agashe from the National Center for Biological Sciences, Bangalore. Dr. Deepa Agashe is a biologist working at the interface of evolutionary biology, ecology, and molecular biology. Dr. Agashe's adaptation lab at NCBS studies evolution of complex systems using diverse tools, ranging from computational analyses to natural or laboratory populations of bacteria or insects. She received her bachelor's degree in microbiology from the University of Pune and her PhD degree in ecology, evolution, and behavior from the University of Texas at Austin. Then she went on to do her postdoctoral work at Harvard University. Dr. Agashe was awarded one of the 3 Science and Engineering Research Board, or the SERB, Women in Excellence Awards 2020 by President Ramnath Kovind. In addition, she has successfully competed for several national and international research grants and fellowships. She serves on various committees and was recently elected Vice President of the American Society of Naturalists. We are happy to have you here, Deepa. Before we start, I would like to remind everybody to keep their microphones and videos turned off during the webinar. If you have any questions, feel free to type in your questions in the chat box. We'll be monitoring them and addressing your questions at the end of the webinar. With that, over to you, Deepa. Yeah, thank you.
Okay, wonderful. Thank you so much by inviting me to speak here. It seems like a really great idea and a great forum to discuss latest developments in science. And so I'm really excited to tell you a bit about some of the work that we've been doing in my lab in the last few years. So let me get going with this presentation. So as the title suggests, I'm going to, can you hear me really clearly? And
I think so. There's no echo so far, and we'll be also be monitoring. Thank
okay,
you.
great. Okay, so I'll be speaking today about the evolution of complex systems and using bacterial translation and our work with bacterial translation as a case study today. So let me get going. The first question that probably many of you are thinking is, what are complex systems, right? How do we define complex systems? And of course, there are as many definitions as many ways of defining complex systems as there are people probably studying them. But I want to just give you a glimpse of the kinds of things that come to mind when we say complex systems. And in biology particularly, what do we mean by complex systems and why do we study them, right? So one of the first things that may come to mind is this sort of seemingly haphazard mess, right? Which seems very, very hard to untangle, to understand what is going on, what are the underlying threads, what if I pull one of these threads, what is going to be the outcome, right? So complex systems, this is of course a piece of art, may be represented by very sort of multi-layered interactions or multi-layered processes going on in any system, and these can be depicted in art as well as science. But I want to discuss another example today from a biological system which some of you may know of, which is fascinating and is sort of one of the exemplars of the kinds of complex systems that people study. And this is this phenomenon called murmurations. So these are Rosey Starlings. I hope you can see the video that's playing. And I saw this congregation of starlings last December around near Pune. And these starlings come in the evening to roost in these enormous masses. And you can see that they form these very interesting and dynamic shapes and patterns in the sky as they fly around for about half an hour before they settle down for the night in some of these trees that you see there. And this is such a fascinating phenomenon, this kind of mass emergence of properties of a collective group that comes about even though each individual seemingly is doing its own thing, right, and taking decisions on its own. So in this kind of complex system, it's really interesting and important to understand, well, what are the kinds of individual level processes that are occurring that give rise to this collective phenomenon? And can we start now beginning to describe what are the factors that lead to this phenomenon and what are the interactions to be able to predict, for example, precisely where this group is going to go in the next second or in the next minute and so on. At the moment, we don't have...
I'm so sorry to interrupt,
Yeah.
Deepa, but someone I think had like clicked on the present now, so I think it got messed up. Could you please go ahead and select present now again? And just a reminder to everyone else, please don't fidget with the settings, and just keep your microphones off and your video. Yeah, thank you.
Okay. All right. Is that visible to people?
Not yet.
One second. Okay. It says my screen is still visible to others. Okay, maybe I'll just stop screen sharing and start again, and that might.
Okay, thank you. Thank you.
Okay, is it working now?
Yes, it's working now. Thank you.
Okay.
And
okay,
yeah.
Great. So
everybody, sorry for the tech issue. Yeah, thank you.
No worries. Yeah, that's fine. So then I think, did I go with the video? People saw the video, or that was lost as well?
I think that was lost. So if you don't mind, yeah.
Okay, sure. Yeah. So hopefully it's visible now, and you can see these murmurations of Rosey Starlings. Yeah. Okay. So I think what I spoke at least was audible to everybody. So hopefully now it makes a little bit more sense what I was talking about. So these are individual birds, like I said, and it's interesting to think about, well, what is this emergent property? How do we describe this emergent property? And how do we start describing what are the layers that and interactions and other factors that feed into driving these kinds of very interesting emerging and complex behaviors? So even though the system seems like it's fairly straightforward, there are, I don't know how many hundreds, how many thousands of birds here, but because each is an individual that's different, the outcome is a little bit unpredictable, and we don't yet know what exactly drives this kind of phenomenon, right? So that's another kind of complex system that biologists are interested in understanding, emergent properties. And finally, my last example in this situation is that of eyes, which is probably, again, one of these really famous examples of what's a complex system. So it's a complicated piece of machinery in some sense, right? And here, I think it's easier to think of in biology exactly what we want to get out of these complex systems or what we want to understand. In many cases, the diversity of eyes or light sensing organs effectively, even though they're all performing at some peripheral level the same function, the precise wavelength which they will detect, precise output is going to be different, right? So the input and the output may vary depending on what animal species we're talking about, but the underlying function is the same. And this is called a complex because there are so many parts, little small parts that have to interact in a perfect way to be able to give the output that is desired. And in this case, the output is something that is the outcome of natural selection or evolution over millions of years of time, right? So as a result of this evolution, that's selecting for slightly different outcomes of the same or similar kind of system to be tuned to the specific habitat in which the organism is evolving, you end up getting this enormous diversity of, so starting with a simple machinery, which is simply a light sensing organ, right? So I want to, this is sort of closest in some sense to the system that I want to understand today in my example, and that is translation, or the process of producing proteins in bacteria. So in this screen, I'm showing you, just as a reminder, all the different components that go into producing proteins from the messenger RNA or mRNA strand, which is shown here in this cartoon. You see a ribosome on the left, which is made up some abstract version of a ribosome, which is going to read the mRNA, right, from left to right on the screen. And it needs tRNAs or transfer RNA molecules to bring in the appropriate amino acids which sometimes may also carry modifications. So the tRNA may be modified sometimes, and these tRNA modifications, as you'll see later, play a big role in the story I'm going to tell today, and they are actually important aspects of translation. We don't think about them a lot, especially when we read about translation in textbooks and so on. But tRNA modifications are really interesting to understand how the genetic code evolved, because they allow a particular tRNA to recognize not only the perfect match between the anticodon and the codon, so read codons for a specific amino acid, but the tRNA modification also allows the tRNA to recognize other codons which are similar, not the same, as the codon that it's supposed to normally recognize based on perfect base pair matching between the mRNA and the codon on the mRNA and the anticodon in the tRNA. Right? So tRNA modification systems are also...
I'm so sorry to interrupt again. I'm sorry, but in the chat box, and we also, I think, the slides are not changing, so I don't know. Now it did. Earlier it was just on the first
maybe there's a lag.
Or
on the first slide, meaning
your introduction slide. So is it on the slide play, like slideshow mode?
No, I'm changing the slides, yes, as we go along.
Okay.
Can you see this slide now?
Now it's on the translation slide.
Yeah, so that's the only slide I've shown so far, really, after the introduction.
Okay. It wasn't changing. Okay, sorry about that.
Okay.
So yeah, if you just click on it as you go along, I think it shouldn't be a problem. Okay.
Yeah, I mean, that's what I was doing. So maybe I'll mention the next time I change the slide or click on something should happen. Okay. And if it doesn't happen, then you have to let me know. I'm not sure what's the alternative though, but okay, all right. Right, so the modification system is important, and there are many ways that errors can creep in the process of producing proteins, right? So now there should be a small animation which hopefully plays. Do you see that? Hello?
No, no.
Okay. Now, do you see anything happening on the screen?
No, it's a schematic of the translation system, but yeah, there's no animation happening.
Okay. Is the slide change now slightly?
Yeah, it's yeah, there's information, I think, after you clicked, so.
Okay, so I'm not sure what to do about this, because yesterday I think you guys were able to see my slide changes quite well when we tested this, so I'm not sure how to go about it. I can stop presenting once more and see whether that changes anything or matters.
Okay.
I think, Deepa, if you have the option play on the top of the keynote, will that work?
That's what I've been doing.
Okay.
So I'm not sure what is going on. So I'll stop presenting and then start again.
Okay.
I wonder if I share my entire screen, that would make a difference. So let me try that this time.
Okay, sure, thank you.
And then maybe that. Okay, let's see. Okay, so now there should be a small animation that happened. Is that showing up?
No, it's not coming.
I mean,
okay,
yeah, it's coming. Yeah.
Yeah. Okay, great, wonderful. Okay, fine. So something is changing now. All right, great.
Okay.
No, it's fine. I mean, I don't know why this was not working. So, okay, great. All right. So now there are many ways of getting errors, right, in this whole system. And I'm sure some of you already know some of this. So one way is that the start and the stop signals in the mRNA may change because of mutations. So let's say now the start and the stop come closer together because of new mutations that occur. And as a result, you might get a shorter protein or a peptide than what you originally intended to make. You might also get errors because some of the tRNAs have disappeared. They have been deleted, let's say, or the expression level of the tRNA has changed. And that also is important because if the right set of tRNAs are not around in the right proportions, the ribosome is more likely to make mistakes because it will incorporate some other tRNA instead of the tRNA that is needed, right? Similarly, if you lose the tRNA modification, people have shown experimentally previously that if you don't have tRNA modifying enzymes working as normal, then the process of tRNA amino acid incorporation is more likely to have errors in some cases and is also likely to be slower in some cases, right? So there's a cost to be paid if you don't have the system working as it's supposed to work in some sense. Finally, in the ribosome, hopefully you see a red triangle now, and that triangle signifies some mutations in the ribosome that could also alter its fidelity, right? So it could either make more mistakes or fewer mistakes and so on. So all of these are the breadth of, well, some of the breadth of the kinds of changes within a system, which may mess with the ability of translation to be accurate and fast, right? And both of these can be problems. So then this very complicated system with so many parts, how does it function and how does it evolve? That's the question that I'm going to try and address. The functioning, of course, a whole lot of people in the last few decades have figured it out, and many people are figuring out now. But the evolution is my primary concern, and I'll talk to you about that more. Okay, so what I want to highlight now is if we pare things down to these few components of translation that I mentioned right now, it's actually surprising for such a fundamental process that is undertaken in all cells in life. There is a lot of variation in different components across different species, and today we're going to focus only on bacterial species, but I'll just mention that there's actually even more variability across eukaryotic systems. So mammalian systems, humans, bacteria, they all have very, very different sets of modifying enzymes, for example, and tRNAs that they use, and different components, right? The core structure remains the same, of course. It's largely shared, but there are lots of very interesting differences that do pop up. Okay, so as an example, the ribosomal RNA, which is a critical component of the ribosome, can vary from 1 to 15 copies, right, across different bacterial species, and that is known to correlate with the possible growth rate of the bacteria. So it's thought that the number of ribosomal copies, RNA copies, is limiting for ribosome assembly. And so that's why if you don't have enough rRNA in the cell, your cell is not able to translate as fast as it should. There's also a lot of variation in codon use. So the kinds of codons that are used in the mRNA, some bacterial species will use a particular codon much more often to encode a given amino acid. Other species may use a synonymous, a different codon to encode the same amino acid, right? So there's a lot of variability in codon use as well. And as you can imagine, that should impose selection for tRNAs to then match with the codon use. So if you look at across bacterial species, the number of tRNA genes in a genome can vary from 20 to 120. The number of tRNA copies for a given type of tRNA, that is a tRNA that has a specific anticodon, can vary from 1 to 25, and that seems enormous, right? For tiny bacterial genomes, 25 is a lot. 25 is, I think, in most cases, just the first methionine, the forming methionine, because it's so critical for beginning of all proteins. But there is still a lot of variability across the same other amino acid carrying tRNAs as well. And there are 25 to 46 types of tRNAs found across bacterial species. And you'll notice here that this number 46 is much lower than the simple naive expectation you might have based on the number of codons that we know of, right? So there should be 60 codons that there should be found apart from the stop and the start. And we don't see that many types of tRNA. So that automatically tells you that tRNA modifications as well as GU wobble pairing is important. So the tRNA modifications themselves, there are many different systems. So maybe I should quickly explain that for those of you who are not familiar with this, tRNA modifications, I am going to focus today on the modifications that occur at base 34 in the tRNA. So counting from the 5' end to the 3' end, 34th base gets modified in some ways that allows that base to wobble pair with other bases that it would not normally be able to pair with without the modification. So it's a chemical modification of the base of the tRNA. There are also other modifications that occur made by other enzymes in other parts of the tRNA, and that I'm not going to deal with today because those are primarily not thought to be involved in codon anticodon recognition, which is the key component of trying to understand translation accuracy today, right? So hopefully this is all working out fine now. Can somebody confirm?
Yes, it's going well.
Hello?
Hello. Yeah, it's
okay,
going
great. Okay.
well. Thank you. Yeah, it's working now.
Okay, so then the main question I want to start off addressing today is, have different components of translation co-evolved over time? And why might we expect this co-evolution, right? So as I explained earlier, we think that tRNAs, as well as the modifying enzymes that act on those tRNAs, I'll abbreviate them as MEs in the rest of the talk, should sort of correlate with each other, right? So if you don't have modifying enzymes that are able to allow a tRNA to wobble pair with other codons, then it's perfect match. Then you need to have many more types of tRNAs, right? Because every codon must have its own anticodon specific tRNA to be read. On the other hand, in modifying enzymes, tRNA can recognize up to 3 other codons, right? And so with 3 other codons, that means you don't need 4 tRNAs, all of which may encode the same amino acid. Only one of them with a modification can do the job. So this kind of co-evolution we expect to find. And we may expect to find similar kinds of co-evolution with codon use as well as amino acid use. If amino acids that are used more often are found, there are some amino acids that are used more often in a cell, like glycine is used a lot. You would expect to see that tRNAs that recognize glycine codons should be also very abundant. And similarly, for codon use, specific codons of, for example, glycine that are used very often in the cell should also be recognized by abundant tRNAs in the cell. So how does this mean? What does this mean for modifying enzymes and tRNAs as an example? Across the phylogeny, so evolutionary showing evolutionary relationships between different species or groups of bacteria. If we imagine a common ancestor on the left here, shown at the root of this tree, and then A, B, and C as different taxa or clades that have evolved over time of different bacteria, we might see differences as follows. Let's say species A has lots of different types of tRNAs, so many different tRNAs, and there's no selection as a result to have modifying enzymes, okay? On the other hand, let's say species B may have lost some tRNAs. There is now positive selection to acquire modifying enzymes, allowing the few tRNAs it has left to be able to decode the whole set of codons. And finally, in species where it's an extreme situation, there are very, very few tRNAs, you might expect that those species have many more types of modifying enzymes, so that that small pool of tRNAs is still able to decode the entire genetic code and all of the protein coding genes in the system, right? So the question is, in this complex system now, can we start to make some sense out of understanding what patterns are there, right? Do we see all kinds of species in the phylogeny? Do we see lots of tRNAs that are consistently gained and lost? What about modifying enzymes? Are they gained and lost very often? And these are the dynamics that we would predict if these systems were co-evolving, right? So we tested this on a phylogeny of bacteria. And we are focusing here now on this phylogenetic component of the work. So this is a bit complicated. Let me walk you through it. For those of you who are not familiar with phylogenies, on the right side of this, it's a tree, and the branches and the tips of the tree are showing you currently existing species. I've labeled the major bacterial groups here. So alpha proteobacteria, spirochetes, some of you may have heard of them. And then as you go backwards, which is towards the right on the screen of the phylogeny, you're going farther and farther back in time until you get to the last common bacterial ancestor, right? So each node represents a particular clade, which is a hypothetical ancestor, which must have existed at some point, from that lineage, a bunch of new strains and new species have evolved over time, right? So that's what we're looking at. And on this phylogeny of over 1,000 different bacterial species, for which we could get genomes available in public databases online, we did an analysis to map the most likely sequence of evolution of different sets of tRNA genes carrying different anticodons, marked on the right there. So you see CAA, CUG, CUU in the right side corner in the bottom. And the gain of these tRNA genes in different branches and different lineages is marked by filled triangles with different colors indicating different anticodons. And empty triangles indicate our best guess where most likely a tRNA was thought, right? And so this is based on our understanding of or guesses about what evolutionary processes might have happened. combined with the data we have for all existing species, that's the tip of the phylogeny on the left side of this phylogeny. And so we can use, we basically fit different models to ask which model of evolutionary process best explains the data that we have in hand right now, right? Which is the current species, because we of course don't know anything about the genomes of the old species which have gone extinct, or the old ancestors. So as you can see, hopefully, there's a lot of change happening in these taxa, right? So in most of the bacterial lineages, you should be seeing that there are a lot of empty triangles that come up of different colors. There are also a lot of solid triangles that come up, right? So particularly towards the top part of the phylogeny, alpha, beta, gamma proteobacteria, you should be seeing lots of empty triangles, which suggests that there have been lots of situations where the tRNA genes have been moved. And towards the bottom, you'll see firmicutes, and those are the cases where a lot of tRNA genes have been gained in more recent times. And finally, if you focus on actinobacteria, which is kind of in the middle, you find that there not much has changed. So the best guess we have at the moment is actinobacteria have retained a lot of the tRNA genes, or most of the tRNA genes, that the common bacterial ancestor was expected to have. I forgot to mention that the last common bacterial ancestor, based on this analysis, is predicted to have had all of the tRNA genes that we're focusing on here today, right? So now the question is, well, what about the modifying enzymes, right? So these are all tRNAs, just a subset of tRNAs that I'm showing you here for clarity. We've done this analysis for all other tRNA genes. So I'm blowing up now, or zooming in on a section of the phylogeny I showed you before. On the right side is still the same phylogeny, a section of it. So going from actinobacteria on the top towards the thermatogae at the bottom. And I tell you this before, but these are all tRNAs that are not modified by modifying systems at all, right? So these are what we call non-target tRNAs. They do their job by themselves without any modification. On the left side, I am showing you a mirror image of the same phylogeny. What we've done here is we've mapped the tRNAs which correspond to the same amino acid, but which are targeted by a modifying enzyme. That means that they're chemically modified, and those target tRNAs, in some cases or most cases, should be able to therefore recognize the codons read by the tRNAs on the right side of the screen, right? So the target tRNAs could compensate for the loss of the non-target tRNAs. And interestingly, we find that the losses that we see, or gains that we see on the right of the phylogeny, are more or less missing on the left side of this phylogeny, right? So what that means is target tRNAs really don't change at all, okay, in evolution. Evolutionary time. Now, I just remind you that the time scale we're thinking of here, from the common ancestor to the tips of the tree, is many millions of years. So it's super interesting that our best estimate, according to our best estimate, across these multiple millions of years, these tRNAs have never been lost or gained for the large chunk of phylogeny, right, on the left. Whereas on the right, they keep getting lost and gained a lot. This suggests to us that the process of evolution of the components of the translation machinery are very dynamic, and we want to understand this more. On the left, what's also interesting to see is that all of these different lineages are predicted to have modification systems in place since the last common ancestor, right? So whenever there's a modifying system that is targeting a particular set of tRNAs and allowing them to recognize multiple codons, you have the flexibility to gain or lose other tRNAs that now become dispensable. That is what this model that we did, this analysis that we did, predicts, right? So that's really interesting, and it supports the general idea that there is some co-evolution happening between modification systems and tRNA genes, right? And this is exciting because it's across the breadth of the bacterial phylogeny. Okay, so now I want to focus on the second part that I mentioned in the previous slides. Have different components of translation co-evolved over time. Now let's focus a little bit in more detail at the tRNA and codon use and amino acid correlation, which I explained already. I want to bring in a third factor here, which is that of growth rate. Now we also expect other people, work from other people, both experimental and theoretical, has suggested that we should expect to find that whenever a bacterium is growing really fast, that is the time when there should be maximum selection for having most sort of rapid translation, right? So we expect that if you're under selection for fast growth, you should be under selection for having many more tRNA copies and a large pool of tRNAs at the ready to dive into translation as soon as you need to produce more proteins, right? And this is largely thought to derive from the fact that if you are in a nutrient-rich condition, you better grow fast and outcompete everybody else, otherwise somebody else is going to outcompete you, right? And by growing fast, it basically means you need to produce proteins really fast. So that's the simple prediction and simple model here. And so we wanted to again test it using bioinformatics, asking what are the patterns that we see here. So remember, like I said, the prediction is on the x-axis a proxy for growth rate. I'm not going to go into the details of what this proxy is for now, but again, we're using existing known genomes of bacteria. So each dot here is a bacterium. And on the y-axis is the total number of tRNA genes. And as per the simple prediction I laid out in the last slide, we expect a positive correlation here, which is what we see, and it's actually quite a nice positive correlation, right? Now let's go a little bit deeper in, right? Say, okay, now what tRNA genes are contributing to this positive correlation, right? So now I'm showing you just examples of three different, tRNAs that recognize three different amino acids, right? Or that recruit three different amino acids. So it's asparagine, cytosine, and glycine. And the copies of those specific tRNAs, that is carrying specific anticodons, are plotted on the y-axis. X-axis is again proxy for growth rate. And here you see that although overall there's a positive trend, there are different amino acids and different tRNAs that are responding differently. So glycine, for example, is responding much more rapidly to change in proxy for growth rate, right? So if there is stronger selection for growth rate, it seems like this is across species. Glycine is one of the important ones that seems to increase really quickly. Maybe cytosine is not so important, changes but not so rapid, right? So now on to ask, well, why is this happening? What is it that's different about these different amino acids that allows different tRNAs to respond differently? And as you might expect, simplest thing is amino acid usage. So we tested for that. And so on the x-axis here, I've plotted amino acid use in highly expressed genes. And we selected highly expressed genes simply because those are the genes which we think are going to generate the most demand for specific tRNAs and specific amino acids, right? Specific proteins to be produced. So as you go along that x-axis, you find that amino acid use is increasing in highly expressed genes. So for example, alanine, valine, lysine, glycine, these are all amino acids towards the right of that plot. And on the y-axis, you're seeing the slope of that relationship that you see in the left, right? So glycine, for example, is high up because it responds really quickly. So it's high on the y-axis. And what this plot shows is interesting. Those amino acids that respond fast, those tRNAs that respond quickly to growth rate change, are also the ones that are used very often in highly expressed genes, right? So this confirms the idea that if there is strong selection, there's lots of demand for a particular tRNA gene, because that amino acid is used a lot during fast growth, then that tRNA tends to have very quickly a larger number of copies in bacterial species that seem to be under selection to grow fast. Now we do the same analysis for codon bias, right? We want to say, well, just like we see in tRNAs, do we also find that codons, which are encoding for highly used amino acids, are they the ones that are also again responding to fast growth? And we don't see that. Okay, so on the x-axis, you see the same thing, amino acid use in highly expressed genes. But the y-axis here is the slope of codon use response growth rate, just like I showed you for tRNAs. And here you see there is no correlation pretty much, right? And if anything, there's a weak correlation at all. So codon usage in tRNA genes respond differently to selection for fast growth. We find that different tRNA genes show variable responses to selection for fast growth. And together, all of these components are now fitting back to the bigger picture that I showed you earlier, that all these different parts of this machinery have to work perfectly together. And it seems like in different species in the bacterial phylogeny, different species have found different optimal points such that the machinery works, but the details of how it works are slightly different in every case, depending on the circumstance, depending on mutations that happened, depending on selection that may have allowed different systems to evolve in different ways. This plot showed up now, but this is just the tRNA plot I'd shown you earlier. Okay, so now the question is, okay, that's one way of asking how do complex systems evolve, is in nature, how have they evolved, right? What are the examples? What is the variation? And now if we can document in what ways they vary, we can understand how they evolve, because ultimately all of that evolution is telling us it's an experiment that has already happened for millions of years in the wild, right? So now how about experiments that we can do in the lab, right? Because then it's really powerful because you can see it in front of your eyes. Okay, so now the question is, can the system compensate for changes? So if I mess or tinker with the machinery, how does the system adjust, right? That's what we're asking here. And here I'll focus on these sort of tripartite relationship. We're interested in asking what happens to tRNAs, do they influence growth rate and codon use, and in turn, does growth rate influence tRNAs and codon use, and so on. Okay, so we're gonna mess with one of these factors at a time. And this is some work that I did in my postdoc. This is just a small part of a pathway responsible for biomass production, bacterium, Methylobacterium extrocans. This is the model system, not important at the moment. What's important is that it has the ability to eat methanol as a carbon source, right? So on the top of this reaction, you see methanol. And one of the key enzymes it requires to do this is called FAE, which is formaldehyde activating enzyme. And the only job of this enzyme, as far as we know, is to take formaldehyde, which is a byproduct of methanol use, and convert it into something that is non-toxic, right? Because otherwise, if you don't have FAE, what happens to these cells is they accumulate a lot of formaldehyde, and it's just toxic, and they die, right? So without functional FAE, you cannot survive. Okay, this bug cannot survive. So because FAE is very highly expressed in the cell, we decided to use it as our model to say, okay, now if we change the codons in that encode the FAE gene, do we see any difference at all? Remember, codon changes are going to be synonymous. They're thought to not matter for evolution, because synonymous changes would make sure that the amino acid doesn't change. And you'll see, interestingly, that's not the case. Okay, so what did we do? I made all these strains with different alleles of FAE, where I introduced a different number of mutations, which changed the codon, but did not change the amino acid, right? And you see wild type here on the right, in the top right corner, labeled WT, which is wild type. And on the y-axis, I'm plotting growth rate, and on the x-axis is amount of FAE produced, protein produced, relative to wild type. Okay, so the x and the y-axis are both going only up to 1 and starting from 0. And all the colored dots that you see here are points representing growth rate and protein of all the other FAE alleles that I produced with different codon usage. Okay. And what you clearly see is that they're all kind of sad compared to the wild, right? They're not doing well. They're neither growing well, nor are they producing enough protein. Given what I just told you about FAE, it's not surprising that if they're not growing well, they're not producing enough FAE protein, or vice versa, right? And so the question was, well, what has happened with this system? I'm not going to be able to go into the details of this because I'm running short on time, but I'll just tell you what we did next, which is we decided to allow them to adapt to this changed codon use, right? And that is going back to the question I posed earlier. If I mess with one aspect of this system, how do the other components react, right? So I've now messed with one aspect of the system, which is codon usage. And we're trying to ask now, if we let them evolve, if we let the system adjust over some evolutionary time, how does it adjust, right? And so we allowed experimental evolution in the lab in batch culture. And what we see is repeatedly, population growth rate increases very. This is happening within 50 to 100 generations in all of the strains. You see that the wild type, which is in gray on top, does not change very much at all. Right? But all the different colors each represents a different allele of FAE with different mutations, and all of them improve very rapidly, almost to wild type level growth rate. So then the question is, well, what happened in them? And I sequenced the FAE gene because that was my first guess that maybe one way that the system may have compensated is to get back some of the important codons, right? To have mutations such that the key codons would go back to being what they were preferred earlier. Instead, we find that every strain had only a single mutation, a point mutation, and across all the strains, this is the collection of mutations that I found, right? So I've marked some of them as synonymous, some are non-synonymous. This is the first 14 residues of the FAE gene that I'm showing you here, starting with MAK, ITK, BQ, so on. Right? And so you see some, which is the orange dots, are upstream of the genes. So they are maybe promoter mutations. I have not verified them yet. But I focused on a bunch of these mutations which are in the beginning of the gene, so the first 5' region of the gene. And I made those mutations back into the ancestor, okay, of the respective ancestor. And that's the data I'm showing you here in this plot. Growth rate relative to wild type and FAE protein relative to wild type. And what you see is the ancestors are in the circles. And as you see from earlier, all of the alleles of FAE in circles, which are colored circles, are all not growing as well as wild type and poorly and growing poorly. But then when I introduce the evolved mutation, remember a single point mutation, in some cases a synonymous change, it is immediately increasing the growth rate as well as the protein production. So those are all the triangles, right? Where now the rest of the genome is the same as the ancestor. The only change is that 1 point mutation. So this is fascinating because the system is completely messed with us, right? Because we expected tRNA mutations maybe, or maybe some codon changes. That's not really happening here at all. So there are very simple mutations that the system evolves, all of which upregulate the FAE expression, but they don't really fix the original problem that I created. They just find a different solution to it, right? So that's what has happened. All right. And then, so what we find here is changing codon use can reduce bacterial growth, but selection does not directly act on codon use because we don't see evidence for that. And finally, changing codon use probably has many other complicated effects that may drive selection, and we don't really fully understand them. So there are many labs since this time which have done a lot of interesting work trying to understand similar questions and address them in different ways. And altogether, it seems like there are many effects that changing codons have on RNA structure, on folding, on perhaps the half-life. All of them may have a role to play in why codons are used. Some codons are used more often and others are not, and why when you mess with them, it's problematic for the cell, right? Even though you think the protein should not change at all. So this is still a very active area of research, and if some of you are interested, I definitely encourage you to pursue it. All right, so then the last section quickly, I will tell you about all of the stuff that I've told you about so far is predicated on the idea that making mistakes is a bad idea, right? That translation errors are not so useful, that you want to avoid them. They're an unfortunate problem that comes up because the system is so complex, but we should sort of keep it low as much as possible, right? But there's an intriguing idea that maybe the errors that do happen can sometimes be useful. And so we wanted to ask, when could that be the case and why, right? And so what we did here is we manipulated the rate of translation errors in E. coli. So again, to remind you, this is the system that we're working with. And I marked here the FMET, which is the formal methionine, which is the first amino acid that is added, the methionine that's added at the beginning of a particular every protein. The methionine that is added in the middle of proteins is different. It's carried by a different tRNA. So in E. coli, there are 4 different genes that encode FMET, and of those we deleted 3, right? So now initiation of protein production is a problem, and this is what we call the mutant for our work, and we compare that with wild type. Another way, so the mutant now has a higher rate of error, also a lower rate of protein synthesis in general, because we've basically made sure that there is a very low rate of protein translation initiation happening. Another way that we manipulate translation error, we introduce canavanine or norleucine into the growth media. These are amino acid analogs which simulate errors in translation, so proteins have mistakes in them. And finally, we used hyper or hypoaccurate ribosomes. These are ribosomal genes which have mutations. These are very well characterized mutations by a lot of other people in the field. And they have shown that these specific mutations in the ribosome sequence can allow the ribosome to be either high fidelity or low fidelity, right? So we use both of these ways to basically start from wild type and then explore wild type level or away from wild type level mistranslation rates and ask what happens in the cell. So unfortunately, I'm going to run out of time now, so I'm going to not tell you about a lot of the details of this work, but it's published, look it up, please. But I'll tell you the most fun part, right, or the gist of it. So what is that? We found that mistranslation increases early survival under stress, right? So we're comparing cells with wild type cells, and with cells that make less mistranslation than the wild type and more mistranslation than the wild type. And we're asking how well do they survive under different kinds of stresses. So we use DNA damage as important stress. So we use UV, hydrogen peroxide, we use ciprofloxacin, which is an antibiotic that damages DNA. And using all of this work that we did pointed to this simple schematic that I'm going to show you here, right? So you have population on the top with low mistranslation, okay, that's your wild type, and your population with high mistranslation rates. So it's making more errors in protein synthesis. Now, if you expose it to some stress, what happens? We see that, and this is based on a bunch of work that we did in the lab, we see that many more cells die if you're not mistranslating at a high level. Okay, so these dead cells are shown here by these dotted circles. And some of the cells, many more of the cells in the populations that have high mistranslation rates are able to survive. Okay, and so early survival is increased. But at this point, if we sequence these cells, we find no mutations at all. Okay, so this difference in survival, which is about twofold, is actually quite interesting because there are no mutations happening at all. There's something else going on. Okay. Then what happens is slightly longer term, the stress continues. This is DNA damage. The survivors now start getting mutations because they're trying to replicate. And as they're replicating, they're getting mutations. There's also DNA damage that is being repaired. There are mutations, right? So now there is going to be some probability of finding a beneficial mutation, okay? In this case, this is ciprofloxacin that we're dealing with. The stress is antibiotic. And so very few cells in the wild type with low mistranslation end up getting the right mutation because there are few cells to begin with, right? So it's a population size problem. On the other hand, with population that has a high mistranslation rate, there are many more survivors. So there are many more of them that happen to get a mutation by chance, right? And now those mutants, because they have the mutation that allows them to survive long-term under the continued stress, they are the ones that now start proliferating, and they make up all of the population that we see after a long time. So after 24 hours under ciprofloxacin stress, everybody has gyrase mutations, okay? And gyrase is a known target gene, which has to get mutations for cells to survive in ciprofloxacin. This is a common antibiotic resistance gene that is targeted and that mutates in response to antibiotic selection. So now at the end, if we look at what mutations are there, which cells are there, there are cells, all cells have gyrase mutations. But populations with high mistranslation have more such cells, and they have therefore greater survival on average. Okay. Why is this happening? Here's again a schematic showing you why we think this is happening. So there is a response in bacterial cells called Sos response. This is a sort of a master response which allows cells to respond to many different kinds of stresses in the cell. And what we found through molecular work is that the Sos activation threshold seems to be set by a number of molecules. And in the mistranslating cells, what happens is because there's a lot of mistranslation, one of the molecules that seems to be key activator of the Sos response is increased already, right? So in a normal cell which is going along, a mistranslating cell, because there's lots of proteins which are mistranslated, it ends up upregulating this protein called LON, which is a indicator signaling molecule, right? And that is already brought the cell closer to the threshold for Sos response. And then when we give DNA damage, the cell is already poised close to the threshold, a bit of LON gets activated more, cells are able to respond. What this response does, the Sos response does, is in the mistranslating cells, it allows faster repair of DNA, and therefore the cells are able to survive, okay? This is our best working hypothesis at the moment. We have some evidence showing different aspects of it. We'd like to get more. We're continuing in the future, right? And so this is just an interesting cartoon drawn by one of the students in my lab, showing these sort of monks who are sitting on the right. The two monks are sort of very angry monks who are already always in a bad temper, right? And on the left are these sort of very calm ones who are, just peaceful and they're not angry. But now when there's a demon and it attacks them, the angry guys are ready with their sharp and with their, curses, whereas the calm ones, they die. And so in this case, the bad-tempered monks are better able to survive the demon compared to the normal cells that we see on the left. Yeah. So, all right. So then I'll end with just highlighting some interesting questions about this complex system. I've shown you, I hope, that translation errors, clearly the whole system has evolved in many different ways to try and reduce error rate. Yet, in some specific cases, we find that the error rate being high is actually perhaps beneficial. So there's this tug of war and this balance under which this very complicated system may be evolving in the long run. And what we want to ask now is, just like I showed you, when we change the codon use, what happens to the system? We want to ask, when we change tRNAs, what happens to the system? Does the codon use change? What goes on? We're finding it out now. We have some early data, but not enough yet to share. What sets the limit of translation errors? What decides how many mistranslation, what is the mistranslation rate that is optimal for a cell, and what sets that limit? And finally, can we infer past ecology from current translation components? So if I know for a new species of bacterium, what is the components of the translation machinery, can I predict under what selection did it evolve in the past? And that would be really fun to be able to do. So I'd like to end by thanking the people in yellow here. These are the people whose work I have mainly shown you today. There's a mix of PhD students in my lab and postdocs who led the project. And Christopher Marx was my postdoc mentor with whom I started the work on codon bias. In white, I have shown you people who have been really important to finish the work that we showed. So Avdesh, Tejali, and Riaz were from the next generation sequencing facility in NCBS, and all the other people are people who advised us or shared material with us for various aspects of the project. This is a picture of my lab, which I think has everybody who has contributed to this project and others. Some of them have left the lab now. And then, of course, on funding sources, without whom we would not be able to do this work. So thanks a lot for your attention. Sorry for the technical problems, and be happy to take questions.
Thank you so much, Deepa, for the wonderful presentation, for talking to us about your postdoctoral work and the work that's going on in your lab. We do have quite a few questions. I'll try to go through them. We may not be able to cover all, but I'll try to pick and choose some. So the first one is, when you talk about allowing the system to evolve and adapt in the lab in response to your design mutations, what time scale are you talking about?
That was about 50 to 100 generations, the evolution experiments. So maybe 2 to 3 months. And the other system I mentioned, the bioinformatics analysis, that was across millions of years of evolution. These are just the species that exist currently. So that's not experimental work that we did. Does that answer your question?
I think so. Yeah. Yeah, I'll go on to the next one. When you mutate a codon in the ORF, do the bacteria alter the cognate tRNA level? If so, what is the mechanism?
Yeah, so we didn't find that was happening, at least not in obvious ways. It's very hard to measure, well, it's not easy to measure the levels of different specific tRNAs in the cell at any given point of time. And particularly for the system that I was using during my postdoc, the methylobacterium system, it was even harder for that system. So we couldn't directly measure tRNAs. What we did measure is we asked whether there were mutations in the tRNAs in the promoter region, for example, which would be necessary to regulate the tRNA genes. We didn't see that. What is known, however, is there are plastic responses. So tRNA genes are regulated. So in a cell right now, if I instantaneously change the codon usage, or I change the cell's stress level or other things, the tRNA genes do change in response to that. It's unlikely that at an evolutionary time scale that change happened, because we didn't see promoter mutations in the tRNAs or any gene duplications or deletions.
Okay, thank you. Another one, can mistranslations be put to use artificially or as an application, for example, to generate desired proteins?
Not the kind of mistranslation that I talked about today. So we deliberately wanted to use a mistranslation method that would be global and that would be unpredictable, because we wanted to ask what happens in a natural system if the rate of mistranslation increases. right? Now, there are specific cases where mistranslation occurs. For example, there are known cases where it happens only under certain circumstances and only at a certain site. So, for example, one way in which mistranslation rates increase specifically is read-through error, right? So there's a stop codon and there's a premature stop codon sometimes, and then that first one is skipped in a normal situation. Let's say that the cell doesn't stop translating, the ribosome doesn't stop there, but in specific instances it stops, right? So basically it produces it's able to produce two different kinds of proteins from the same mRNA, and that seems to have evolved to be programmed in a particular way. And there's some evidence for this. So that is a case where you know exactly where your mistranslation is going to happen. But with the kinds of things we use, it's a bit random, it's stochastic, and we don't know exactly what is getting mistranslated and where.
Okay. Another one is, what is the rate and effect of suppressor mutations in survival after missense mutations?
Not sure what you mean by that, simply because once you get mistranslation or missense errors, it may not impose a sufficient cost to the system that there will be any suppressor mutations that arise. Remember, these are mistranslation errors, not mutations that come up, right? So if there's a few mistranslated proteins in the cell, many times the cell is fine, it's going to be all right. In our case, for the mistranslating strain where we removed a bunch of the initiator tRNAs, the FMET tRNAs, in that case, we know that the cell grows slowly, right? So that population, that strain is slower to grow compared to the wild type. And there is a big cost. And we've done some evolution experiments recently in the lab now where we've asked how do they adapt and evolve to this fact of having a smaller, lower growth rate. And we sort of don't have all the data there yet. So I'll tell you in a few days there. But they don't evolve very quickly. They don't sort of come up instantly.
So we have questions from both Google Meet and YouTube, and I'm asking from both platforms. So there's another one. Can mistranslation of protein in bacteria cause dormancy or change in metabolic growth?
It can, sure. Depends on what gets mistranslated and how much mistranslation is occurring. So I guess I can't give a straightforward answer because it's unpredictable.
Okay. And there's another one. It says, compare and contrast the advantages and limitations of studying systemic complexities at the microscopic versus ecological scale.
I don't think there is anything useful I can say there. I mean, because clearly both are important and interesting, and both have their own benefits and problems or limitations, right? So I feel like that comparison is not particularly useful because your questions or your goals are going to be inherently different when you're trying to understand an ecosystem versus trying to understand what's happening inside a cell. So I would say whatever you find interesting, you should just do
that. We'll take about 2 to 3 more, and then we'll try to wrap up. Another one, if there are translational errors in proteins involved in bacterial RM system, can there be survivors under stressful condition?
I don't know. I'm not sure why I might or might not predict either way. I guess it's a simple answer without thinking about specific stresses or specific kinds of RM systems will be modified. So don't have a useful answer, sorry.
Okay. Another one. Does the tRNA gene variability differ among bacterial species?
Yes, so the tRNA gene content or the tRNA gene pool varies a lot across bacterial species. Maybe this was one of the few slides at the beginning which somehow got missed in the full technical drama, but it does vary quite a lot.
Okay. And we'll take one last question. Does mistranslation work in the same way as mutation? Because in the end, we get different proteins. But why do cells survive better in that case?
No, so what's going on is it's not the same. It's quite different, because with mutations, the progeny is going to inherit that mutation, right? So once a mutation happens in the DNA, unless it is repaired, it's going to propagate to the next generations. So once a mutation happens, it's kind of a permanent effect. Of course, it can be fixed, but it's permanent, more or less, right? If there's a mistranslation event, A happens only in a few proteins, right? And B, it is not inherited by the daughter cells. Of course, the daughter will inherit some of the same protein molecules from the mother cell, but it will quickly regenerate its own proteins. And if its DNA is not mutated, the rest of the proteins it's going to produce are going to be normal proteins, right? So that there's a really big difference in mistranslation errors versus mutations. And in our case, what we showed is part of the reason the mutation is problematic is because of the scale at which it happens. So there's not mistranslation happening in the cell, then the cell suffers, right? And in our specific case, it was beneficial because with mistranslation, under normal conditions, the cell still suffers, okay? It's still growing slowly. But what happens is under stress, it has an advantage. So it doesn't have an advantage under normal conditions. Under normal growth, the normal cells do much better than the mistranslating cells. Mistranslation is only beneficial under stress because it is able to trigger the SOS response, which then allows DNA to be repaired really fast. And that is the key thing. And because DNA repair is important in that context, under that stress that we introduce, the DNA damage, that is why the mistranslating cells get a benefit.
Thank you so much for answering the questions. And everybody,
Sure.
Sorry we couldn't take all your questions, but do feel free to look up Dr. Deepa Agashe's work on her website. It's called the Adaptation Lab, or follow her work on Twitter. And you can get her contact information and contact her in that case. Deepa, thank you very much for being here and talking to us about your work. I'm sure the initial minor hiccups don't come in the way you explained it, and everybody was in sync with that. So thank you again very much for talking to us. With that, I'll actually like to turn it over to Shaunak to actually introduce or talk about our speaker for next week. So over to you, Shaunak.
Yeah, right. Thanks, Anjana. Thank you, Deepa, for taking the time to speak in our webinar series. And we do apologize for the technical glitches at the beginning. But it went really well overall, and for such an insightful talk on bacterial translation.
Thank you very much. Thanks to the audience. Yeah.
So thanks to you and everyone for joining. We'll be back again next week, same time. We are delighted to have Professor Dhrubajyoti Chattopadhyay as our next speaker. We'll start the registration soon, so keep an eye on our YouTube channel and Facebook page. And as always, if you have any feedback, please feel free to send us an email. So with that note, thank you once again to Dr. Deepa Agashe, and goodbye, everyone.